I will invite our colleague Dr. Sonu Gandhi to formally introduce our next speaker.
So again, a very good afternoon to Dr. Priya ma'am and our director and Dr. Sharmila ma'am. So it gives me really a immense pleasure to introduce Dr. Sharmila Bapat. Dr. Sharmila Bapat is a scientist at the National Center for Cell Science, NCCS Pune. Madam leads a research group in ovarian cancer biology, including the ovarian cancer stem cells and epigenetic changes in ovarian cancer and network modeling in ovarian cancer. Madam has received the numerous awards and prestigious fellowships for her scientific contributions and excellence. To name a few, she was honored with a National Women Scientist Bioscientist Award in 2008, the R.M. Tiwari Research Oration Award in 2010, the Cytometry Society Award, PCS Award in 2015, Fulbright Nehru Academic and Professional Excellence Award in 2015, Outreach Lecture Fund Award of United States International Education Fund in 2016, and very prestigious Tata Innovation Fellowship for, from the Department of Biotechnology in 2017, from ICMR, the DHR International Fellowship in 2015. She is also an elected Fellow of Indian Academy of Sciences, Bengaluru, and the National Academy of Science, Allahabad. Madam is also a council member of Indian Academy of Sciences, and on the several groundbreaking contributions, Dr. Sharmila's team developed a method to determine the cell profile of the tumor, enhancing the effectiveness of cancer treatment. Her efforts in elucidating the basic biology and regulatory mechanisms have revealed the tumors as dynamically evolving systems that continuously acquire phenotypic and cellular, molecular, and functional heterogeneity. And I can conclude with that excellent achievements and contributions of Dr. Sharmila Bapat that trust your hard work and it's unlocking doors you can't see. So with this, with due respect, I invite Madam to share the thoughts on plasticity in biological systems.
So thank you for that really kind invitation. And at the outset, I think I would have a lot of warm greetings for the entire NIAB family, the past members, the present members, and also the future, because I think the foundation of this institute, as has been outlined to us by Dr. Sudhir Muzumdar and Dr. Taru Sharma, indicate that a sound foundation is going to actually catapult you into a future success. So again, I'm really fortunate to be here to hear Dr. Priya Abraham, who's literally a neighbor in Pune, but somehow I have never heard her talk. And now I think the challenge is up to me to keep the talk as interesting as hers was. But I think even I'm at an advantage because, you know, the research I do is actually it's for me. And one of the reasons I admired her work is that her research has implications in the present tense, in the immediate future, whereas we as simple researchers who have independent groups in research institutes look at problems which fascinate us, which fascinate our students, and the success often is dependent on how involved our students also get into what we think should be the future of, say, any disease, which is ovarian cancer in the case of my lab. So why did I choose to talk on plasticity? I wasn't sure how I could talk only on cancer biology in this institute. And in the long run, I think plasticity is a phenomenon that actually encompasses all kinds of biological systems. Classically, plasticity and elasticity is used to describe the physical state of mostly non or inanimate matter. But in biological systems, plasticity is also an ingrained part of evolution. For example, several species, insect species, larval species, as well as several other vertebrate animals, actually have evolved during the evolutionary trajectory to have a kind of a phenotypic plasticity that will help in their camouflage, that will protect them from predators, and that will give them a survival advantage in the long run. So you have a caterpillar, you have another chameleon, which is so well camouflaged in the background. And again, there's a lesson for us to learn. Classically, the army fatigues, as they are called, protect soldiers by just providing a similar type of a camouflage. And this is a learning curve that goes on forever. So some of the features are always actually fixed into the evolutionary pattern, whereas sometimes, even nature tries to test out several features before making it a permanent fixture in the evolution of that particular species. For example, this particular species, which is the emerald moth, has a larva which carries these purple colored structures on its back, and those are quite similar to the flowers that it feeds on. So that is such a wonderful lesson for us about regarding survival. And later on, the emerald moth, which actually lives on a very young bushes, can camouflage very readily into this. You also know about chameleons, in which you have a changing phenotypic colors. And these are really not ready for evolution to allow them to fix this particular trait, probably because they change their surroundings very often. That necessitates it to have this type of flexibility. And actually, there's a huge group that is studying this type of a flexibility and trying to devise bionic adaptive camouflage material, which would similarly change on receiving a certain amount of light, a certain wavelength of light, and so on and so forth, which will again give armies a bigger advantage in their camouflage. So enough about the external appearance or the external phenotypic features that actually suggest that plasticity is really an important aspect of nature. Plasticity is also important for tissue and organ homeostasis. It occurs at all levels of a molecular cell, of the molecular regulation of a cell, right up to how it should function in an organ. So these are some of the issues that we have been looking at in our lab at NCCS over the last 20 years or so. But so to outline some of the lessons that we have learned over there is that the phenotypic plasticity actually can translate into a functional plasticity, which will allow or give an advantage to long-lived individuals and the organs. So, for example, when you look at an organ, you actually see that it has different types of cells, which are some of the cells are specialized and perform very specific functions. They also have a defined lifespan. So after that lifespan, you have an entire regenerative process that is kicked into place, and that regenerates these cells. Now, what is the advantage of that? Firstly, primarily, you allow them to actually overcome mutations, because one knows that if there is an extensive accumulation of mutations, it can slowly lead to transformations. So if at all there are some mutations that occur in the differentiated cells, they'll actually just be limited by their specific lifespan, and they'll die after that specified time period. Now, this type of a hierarchy that defines the regeneration of organs. You can't hear me? Okay. So there's a defined hierarchy. So starting off with, say, the rarest populations of cells within any organ are the stem cells. Now, this normal stem cell is a very innocuous looking quiescent type of a cell that is present in very specialized locations within the organ, which are called as the niche. And the niche actually is composed of two types of components. It will have an acellular component, which might be the ECM that is present in that particular location, or it will be surrounding by different types of cells, for example, the stromal cells, or it could be the vascular cells that will try to maintain them in a state of dormancy. Now, these dormant cells or quiescent cells are then triggered by microenvironmental signals. Say, when there is a requirement where there's a depletion of some of the specialized cells within the organ, the signals will be sent to these stem cells and they will enter into a state of division. This is extremely slow cycling initially, and it will actually give rise to two types of cells. One is a cell that is similar to itself, a stem cell that may go back to the same niche and stay in a dormant state to again perform the same functions later on. The second cell actually goes on to become the workhorse of the regenerative process. It may undergo a few asymmetric divisions whereby it might generate new stem cells. For example, in conditions, it has been seen that in the survivors of the nuclear bomb, there were a lot of stem cell activity and a buildup of stem cells in several organs in the normal tissues. And this was actually reproduced extensively in experimental model systems. And one understands that even progenitors actually can undergo these asymmetric divisions if there is a need to expand the intrinsic stem cell pool within the tissue. Now, it then starts building up the progenitors. Initially, the first level of the early TA cells or the transiently amplifying progenitors are actually slow cycling, but later on they pick up, they generate a critical mass of progenitors. Then these tissue-specific progenitors will go on to form the terminally differentiated cells, depending on whatever is the need at that particular point of time. And hence, this effectively, again, these terminally differentiated cells are marked by a defined lifespan, and they will die out after their life is over. Because they cannot multiply, they will only perform the specialized function in the location wherein they are actually present. So this type of arrangement has several advantages. As I said, you have a distribution of labor, so as to say, with specialized cells performing specific functions, which enhances the efficiency of their working within the lifespan that they are present in the organ. And again, you do not allow mutations to occur or keep on accumulating in the tissue. So actually, one would be surprised at the number of genomic changes that one finds in tissues, and mutations are actually rampant. But the reason that all of us don't end up with cancer is because these are actually self-limiting and they get diluted out. So that is very nice to know that the body itself has this hierarchy which takes care of us in the long term. Now, this is something that a British developmental biologist had perceived the regenerative process as. Waddington was his name. And he said, according to him, a stem cell is a kind of a naive cell. It doesn't know what it's supposed to do. And depending on the microenvironmental signals, it can go within the landscape of the organ that it is present. It will just slowly roll into the groove in which it is meant to regenerate. Now, this was a very simplistic opinion of the regenerative hierarchy. But what it importantly actually emphasizes is that stem cells are usually unipotent. Rather than stem cells, I would go on to say that he meant probably that progenitors are usually unipotent. They will not suddenly decide one day to make a hepatocyte, the next day something else, or hematopoietic, some of the differentiated cells, a third day something else. They have something called as a lineage commitment, and this is usually a unipotent type of a regenerative capability. Now, over the last few decades, actually what people have been looking at, and most of the work was initiated in the case of hematopoietic stem cells, what people found is when you actually isolate these very early hematopoietic or the primitive hematopoietic stem cells, which are likely to be the most capable of regeneration, what you do see is that they can also trans-differentiate. So these hematopoietic stem cells, for example, will not only generate the lineages, the myeloid lineage or the erythroid lineage, but they can also trans-differentiate and give rise to as diverse cell types such as neurons, the hepatocytes, and so on and so forth. So this was a phenomenon that was found, and that reflects a kind of an inbuilt plasticity, but it also emphasizes the role of the local niche in maintaining the stem cells as a unipotent capability, although this now enhances the capability to a multipotent level. So trans-differentiation again is it can be achieved in two ways. You have directly the trans-differentiation signals. For example, when these stem cells are given specific growth factors, they can actually be forced to differentiate into a specific lineage. And this has several implications in treatment of neurodegenerative disorders. And there are several cases in which this type of therapy has been achieved. Another type of a trans-differentiation is where the differentiated cell actually kind of reverts back into a less differentiated state, and then it can go ahead. It's like a car which is going in one direction. It goes back, it takes a reverse direction, and then it takes a different road, and then there are a plethora of roads that are available to it. And it can, all those different roads actually indicate the different lineages that are possible for it to differentiate. So around 20 years ago, one started expanding this type of understanding of regenerative plasticity from multipotent to totipotent. You had the embryonic stem cells which came into the picture. These were derived essentially from the inner cell mass of the blastocysts, and these themselves were pluripotent, and they could regenerate the 3 germ layers. So when they were brought in culture and expanded, it was thought that this is a fantastic way of actually achieving a large-scale expansion of cells which are pluripotent and which would be useful in treating several diseases. Now, this was actually a very promising area, but although it had two types of setbacks. One was there were ethical, moral ethics issues that of harvesting embryos simply to generate or harvest these type of cells. And secondly, there was always an uncertainty that these embryonic stem cells have a high tendency to give rise to teratomas. So that necessitated a very tightly controlled therapeutic regime wherein these could be actually used for therapy. Now, I think the world has been revolutionized with the induced pluripotent stem cells, which have come up in the last 15 years or so. And this is a kind of a feed forward type of a direction wherein you start off with differentiated cells, and of the 21 different transcription factors that Yamanaka and his colleagues screened, they arrived at a cocktail of just 4 that can be transduced into differentiated cells and to achieve an entire somatic cell reprogramming into a less differentiated cells. So this is actually where you impart to a differentiated cell a high level of regenerative plasticity. And this is where the future actually is now lies in case of therapy. I'm not going to delve on the somatic cell nuclear transfer because I know I'm sitting in an audience which has such a lot of expertise. I see Dr. Muzumdar nodding at his head. So I definitely don't want to step on anyone's toe, and I think I would learn more from you people in this case. Now, as I said, that the iPSC model was something that was brought about by human intervention. So were the embryonic stem cells. So that has led one to think about, does dedifferentiation occur? You know, the whole theme of organ function is that the different components of an organ have their own cellular identity And this is a very rigidly maintained identity. It's an extremely complex molecular program that maintains that cellular identity, which is necessary for it to perform those, for an efficient working of that particular organ. Now, when you think about how an organ gets deregulated and you have several diseases, one of the diseases that one comes up, there are definitely something in the entire hierarchy that is perturbed. Now, earlier one had also seen that when there is injury, you have some cells from the periphery that are actually of the injury that may undergo dedifferentiation, and they move into the area of the injury and try to actually overcome, or it's a self-healing process. So in this context, the process of embryonic, epithelial to mesenchymal transition came to be studied with a huge intensity. Now, the EMT program, as it is called, is actually a developmental program. It is studied extensively in development in several features. For example, during gastrulation, it is actually not a dedifferentiation program, but it is more for migration of the cells or the conversion of the cells into a fibroblastic or a mesenchymal phenotype, which will allow them to actually invade and migrate to the different specific locations within the embryo, wherein they will actually move on to differentiate and give rise to the self-fate decisions of the embryo at that particular point of time. Another type of slightly dedifferentiation event occurs during neural crest migration, wherein the cells of the near the primitive streak actually become mesenchymal, and then they will go on to give rise to the neural crest. In the adult context, as I said, wound healing is one excellent example wherein EMT comes into play. You have the fibrinogen, which creates a fibrin clot, and then the growth factors, especially EGF and so on and so forth, that are secreted actually mediate the epithelial cells to break down their cell-cell junctions, get converted into a mesenchymal shape, and then move into this particular wound so as to fill it up. And then once the wound is filled up, you have a scar tissue that is formed that signals for the process to stop or a MET to occur. That is, if there are any excess fibroblasts that are generated, you have some of them getting converted back into an epidermal cell. So this is a very nice, flexible or a plastic situation wherein you have a EMT that steps in and the reverse process of MET that goes on to complete the process. EMT slowly also came to be realized to be a developmental program that was captured by cancer cells for their metastasis. And actually, I'll go on to talk about this much more. Now, just in the last few years, a certain phenomenon called as Palmigonosis has actually received a lot of attention because it seems to be the first dedifferentiation program that actually may be occurring only in adult tissues. Now, this is, in fact, it was on the cover of this particular issue of Developmental Cell, and it seems to be the two sides of a coin in which you have differentiation or dedifferentiation. Now, what is this program? It's really a very fascinating program, wherein it seems to be an autophagy dependent program. It actually has a very specific, it's a very organized and pathway which has three steps in it, and each step is actually defined by specific checkpoints. So in the first step, the differentiated cell actually undergoes specific degradation of the differentiated cell structures that are characteristic of that particular cell. In most of these, you have DDIT4 that actually will try to inhibit mTORC1, and by doing this, it also activates several genes that are involved in autophagy that will start the basic degradation program. In stage 2, you have a rapid depletion of DDIT4, but the process of repressing mTORC1 is actually taken over by P53, and this continues until the time the entire degradation process is completed, followed by which you have upregulation of IFRD1, which will start inhibiting P53, thereby allowing mTORC1 to be de-repressed, and its upregulation or its expression actually then goes on to license these cells for a re-entry into the cell cycle. And thereby, from a differentiated cell state, you move into a progenitor-like state, which can actively cycle and give rise to fresh cells. Now, this is a very fascinating process, and it has been observed in several organs of the GIT, essentially. For example, in the stomach, during gastritis, you have a large number of cells that are damaged, especially these cells at the bottom of the gastric gland. And what happens, and especially in Helicobacter pylori, you have gastritis which steps in and does a lot of damage. And it was in such situations where the palinogenesis was actually observed. What happens is that what was actually noticed was that in patients who actually recovered from the gastritis had the expression of SPEN, which is a polypeptide that is associated with metaplasia. And these SPEN tissues actually somehow gave rise to a regenerative program, which then which resulted in a kind of a healing process. The same is also seen in case of the pancreas, wherein you have the acinar cells that may be damaged under certain conditions. And these, if they undergo ADM or acinar to a ductal type of a metaplasia, then you have an entire wound healing process that is kicked into place and a regeneration that occurs. One feature that is associated with palinogenesis is that you have a complete metabolic shutdown that in some way complements complements autophagy and allows these cells to reacquire the expression of the progenitor gene expression networks, and that allows the entire cycle to progress further. And then you have, again, this particular graph shows the examples of pancreas, the ADM and the SPEM in the stomach, which may undergo redifferentiation, wound healing, and thereby a normal organ homeostasis is then achieved. Now, the flip side of this is that when you have metaplasia, it also makes one prone to transformation, and you might have some of the transformation networks that may be kicked in, and you would get a state of cancer. So these are the palinogenesis is currently now being, it's just 4 years old actually. So there are a lot of questions in this particular mechanism of dedifferentiation, but it has also been identified in some of the cancers that are associated, especially in pancreatic cancer, as well as in gastric cancers. Now I'm coming back to this particular regenerative hierarchy, because that is where we started understanding how tumors also regenerated themselves. In the early days, when the leukemic stem cells were actually identified in Professor Dick's lab in Ontario in Canada, what was noted by that particular group was that the leukemia was actually characterized by a regenerative hierarchy that was similar to that of the normal hematopoietic stem cells. So we started thinking that would this also be a possibility in solid tumors, and especially the model we were choosing was ovarian cancer, which was a very aggressive disease, and it has a really bad prognosis for a majority of patients. So is it that the aggression of the cancer is defined by the number of stem cells in it? So initially, it was all hypothetical initially, that since the stem cells are the longest lived cells within any organ, they might actually be the site wherein you have accumulation of mutations. And then later on, at some point of time, when the threshold of mutations is acquired, they may tip over and move to a transformed state. So in maintaining the normal organ homeostasis, they actually get transformed, which is something that people are talking of in the context of palinogenesis also today. But later on, it was realized. So you have a stem cell and then it will create a tumor. in the same regenerative hierarchy that is there in the normal organs. However, sooner or later, people also arrive at a suggestion that dedifferentiation could also result in some of the progenitors and the differentiated cells acquiring a stem-like state as it came to be thought of. And this is especially important in case of leukemias. For example, in several leukemias, you know that the progenitors, they are arrested for differentiation, that is, they cannot differentiate further into either of the lineages, and there is an accumulation of blasts as it is caused. And the accumulation of blasts, that is the progenitors, also does not allow the blood to perform its optimal level of functioning, and hence the regenerative system actually keeps on pushing the generation of more and more progenitors, which it believes that it could give rise to differentiated cells. So this becomes a kind of a vicious circle, and thereby you get blast accumulation and very poor functioning of the specific lineages that are affected in this particular case. And again, you have a less number of terminally differentiated cells that are formed. So you have a growing mass of cells that is incapable of undergoing cell death, actually, so as to say, and it keeps on growing. So this is something we have postulated, that these are the three specific parameters that a cell must actually acquire for it to be recognized as a cancer stem cell: dedifferentiation, self-renewal or differentiation arrest, and transdifferentiation. Now, this is something that is now reflected as a hallmark of cancer just earlier this year. So we have been wondering why it was never called a hallmark of cancer. When Hanahan and Weinberg, way back in 2001, set down these set of unifying principles that define a transformed cell, and which are actually, it's almost become a Bible for all cancer biologists across the world. We wonder why cancer stem cells never figured in their own work or in their own principles. Both of them and in their independent labs have been working with cancer stem cells, but they never figured it out. But that they never actually gave any importance to the fact that you have these stem-like cells within tumors, which actually drive the disease and make sure that it becomes difficult to contest the growth of these cells and their metastasis in the body. So according to Hanahan, he has actually just outlined the same three principles that we had identified earlier, and he talks about dedifferentiation. I can't even read it from here. Transdifferentiation and must be block differentiation, what we call as a differentiation arrest. So he also talks about it. It's being important because this type of these parameters actually define phenotypic plasticity, which permits evasion of the normal homeostatic mechanisms, whereby a tissue is going to be limited as far as generation of a critical mass of cells is involved. Now we have, of course, in the meantime, moved on to try and define phenotypic plasticity in metastasis, because for us, when we work with ovarian cancer, it's metastasis that actually kills the individual. And because there are very poor mechanisms by which ovarian cancer can be detected early in its course, we thought that we should have a better management of metastasis. We have been working with a lot of clinicians, and they actually came us, it was a kind of a speculation on their part in one of our meetings that even the specific subtype in ovarian cancer we work with actually presents in different ways within patients. Some of them actually come to the clinic with a huge amount of peritoneal ascites, which is enriched with tumor cells. And it is these tumor cells which have a lot of stem-like features. On the other hand, the same histological subtype of ovarian cancer often presents with a poor amount of ascites, but an equally aggressive disease that will not metastasize much, but in fact, it might be more aggressive and give rise to, and is associated with a higher mortality. So this was a conundrum for us. And we had been wondering about what exactly is going on. We've been working with a lot of tumor samples of these, and this is And we actually realized that it is the mechanism of metastasis that is likely to be different. So this is something that we had also seen in the peritoneal ascites, that you have tumor cells that present not only as single cells, which are the result of EMT. You remember I talked of EMT as a process that generates single mesenchymal cells, which can very efficiently invade through the basal matrix and move on to invasion program of their own. On the contrary, we also found that there were always groups of cells that seemed to actually just detach from the primary tumor and be passively shed into the peritoneum. Such type of cells invariably get lodged onto the omentum, wherein they form their secondary sites of metastasis, and from there onwards, it's just a passive kind of a distribution. Now, this was really surprising because the whole world talks of EMT as the only mechanism of metastasis, whereas for us, the clinical inputs seem to suggest that it is not just EMT, although EMT is something that we have been working with for a long time. And as you can say, we had been working with EMT alone way back since 2005, where we showed that two transcription factors called SNAIL and SLUG, or SNAI1 and SNAI2, are actually associated with ovarian cancer, and their expression correlates with very poor prognosis for most patients. These mediate these effects. As you can see, this is a mesothelial cell which expresses features of both the epithelial as well as the mesenchymal cells. And this is the classical wound healing assay that is often used to find out the migratory capabilities essentially, but it also reflects on the invasive capabilities of a certain cell. Working with EMT also showed us how cells that undergo EMT reacquire a stemless-like feature. Now, when we were working with some of the cells that are capable of undergoing EMT, we found that under conditions of stress, SNAIL and SLUG, which were the two different EMT-mediating transcription factors, they reorganized their global program. So what we were looking at are the modulation of the global target. As you know, most of the transcriptional factors act by the co-activation of being co-activators in a transcription program. SNAIL and SLUG are actually transcriptional repressors. They recognize EBOX in the promoters of genes, and they mediate repression of those specific target genes. So what we noticed that under conditions of stress, SNAIL and SLUG were actually upregulated, but they lost a lot of their original targets and went forward to activate actually two programs. One was a migration program, which allowed them to escape from the site of the insult. And the second was acquisition of several of the self-renewal molecules over here, which allows them to acquire a stem-like state. These cells mostly revert back into a state of dormancy, and from where they acquire the stem-like state. Once the insult, the environmental insult is lost, these then give rise to a recurrent tumor. And very often this tumor is much more aggressive than the primary tumor. So this was a very nice association we had identified in 2009. Now, we also found that this concept of EMT is changing from being just a biphasic state. You have the epithelial state and you have the mesenchymal state. So earlier it was thought, okay, if epithelial cells become mesenchymal and mesenchymal cells then revert back to an epithelial state. Slowly it came to be realized that this is not going to be just a biphasic process, but there are several intermediates that are also generated during this process. And then what is the role of these intermediates? So then we started looking at some of the transcriptional networks. We've been doing a lot of RNA sequencing and systems networks analysis. And we came up with a set of transcription factors that seem to be driving the different clinical subtypes of the same disease actually. Although we did not have a very, we just indiscriminately took whatever samples we could get, and then we were looking at the profiles across these. Now, in today's context, it would be easier to do with single cell sequencing. But at that time, there was no single cell sequencing. So we were just doing bulk RNA sequencing. But through that, we actually identified this network of transcriptional factors that was present in all the tumors. And we went on to see that actually SLUG is a negative regulator of some of the other transcriptional factors. So that meant that probably SLUG defines one mesenchymal subtype of high-grade serous ovarian cancer, whereas some of the others are going to be likely to drive an epithelial subtype because it's a direct negative correlation. It could be epithelial subclass. We went to investigate this type of a phenomenon, and what we came up with was an entire spectrum which represents the plasticity of the tumor, actually, of this particular tumor. So you had cells that were purely epithelial. They upregulated all the classical epithelial markers, and in addition, one specific transcription factor called as TCF21. Then you had the intermediate epithelial cells in which you had acquisition of some of the features of mesenchymal cells, but not all of them. The EM hybrid, as is now actually called in text, in literature as well, which actually is the most plastic state. It can tip its trajectory to revert back to an epithelial state, or it can move forward into a mesenchymal state. So this is actually the most refractory cell of all these different subtypes, and which is even drug resistant. Then you have the intermediate mesenchymal cells. You have an upregulation of the mesenchymal program in these, and the mesenchymal, the terminally committed mesenchymal state, cells, in which the tumors themselves are also actually express mostly the mesenchymal markers. Now, increasingly, we find that these mesenchymal cells, tumors, are actually the ones in which the host immune system is still very well active. So there's a crosstalk of these mesenchymal cells within the tumor, the ECM that they generate, and the type of immune cells that they can be recruited. So we have actually identified that it is this subclass of tumors which is likely to have a better prognosis than any of these. And that emphasizes that we need to understand this plasticity even further. We actually did a lot of assays trying to identify the various functional manner ways in which each of these subclasses are likely to be different. And this is just one of the wound healing or the migration assays, which indicates that the mesenchymal cells actually have a very nice, very fast mechanism whereby it can move into the wound and fill it up, whereas the epithelial cells will even fail to fill up the wound even at the end of 144 hours. Now, more importantly, while this this type of an assay was enough for us to know that epithelial cells do not migrate well, while mesenchymal cells do migrate well. What we realized was from a live cell imaging, when we did this, the epithelial cells actually, I don't know if you can run this. Yeah, if you see this, the epithelial cells actually do not move into this wound. This is the size of the wound. They actually just continue to proliferate, and then they will try to fill up whatever space gaps are there in their periphery. On the other hand, we identified that the intermediate epithelial cells, or the IE cells, they undergo what is a kind of a sheet migration, which is a very nice cooperative way wherein the cell-cell adhesion junctions are not actually lost, but the entire panel of cells actually moves ahead as a sheet and goes on to mediate wound healing. You had the next type of wound migration, wherein you had EMT. I think all of you must have seen individual cells that detach from this group of cells, and they move into the wound and they rapidly fill it up. So in this, the IM and the EM, that is the EM hybrid, and the intermediate mesenchymal cells actually perform both these types of things, wherein they move as sheets as well as an EMT. And this is the most efficient of all the modalities of cell migration and wound healing. On the other hand, you have just the EMT program that is brought into place by the mesenchymal cells, and it by itself is actually not a very efficient way of wound healing. Now, this is actually in direct contrast with what literature says, with the EMT being considered as the one and only mechanism of wound healing. But we really had this type of a thing that we identified based on the clinical inputs. And at the transcription level, what we found is that the epithelial identity is really a very strongly maintained cellular identity, and TCF21 actually inhibits slug expression. Now, this we actually did. Sagar was a student in the lab who did a very fascinating experiment. What he tried to do was he tried to knock out TCF21 from the epithelial cells using CRISPR-Cas, and then he try to express slug. What he found that these epithelial cells would undergo apoptosis rather than get converted into a mesenchymal state. And this is the uniqueness that one sees in ovarian cancer. On the other hand, when he tried to knock out slug in the mesenchymal cells and express TCF21, that was readily possible. And in fact, these cells which actually allowed the expression of slug, which is a EMT, which allowed the expression of TCF21, in fact formed very large spheroids. Now, spheroids are three-dimensional structures of tumor cells, and the uniqueness of ovarian cancer is that peritoneal ascites actually has these spheroids that are present in C2. So the peritoneal ascites in some of the patients which have the mesenchymal subclass of tumors actually have these spheroids. So this suggests that the epithelial state or the epithelial tumors are extremely rigid, whereas the remaining states are very plastic. And okay, this also was a kind of a mathematical modeling we had done. There was a lot of information. There was a lot of learning from those studies that we did. Now we went back to our clinicians. Our clinicians said that, okay, this is all very nice as a basic project, but do you really think that this is what is happening in C2 in the patients? So again, we went back to the patients. We took the tumors. We did single cell. We did RNA sequencing. We did what is called as weighted gene correlation network analysis, and we found that there were modules of correlated genes that allowed us to define three different subtypes in the patient tumors. One was a strongly epithelial subtype. The second was a strongly mesenchymal subtype, and the third was a mixture of the two. This one had an entire program that was driven by TCF21. We had three different types of modules that were upregulated in these. And of course, we did an entire dissection of the different functionalities that these modules defined. While the mesenchymal subtype was dominated by EMT, ECM. And as I said, that you had the immune cells that were also infiltrating into these, which suggested that this subtype had a better prognosis than the epithelial subtype. Whereas the current literature actually suggests that the mesenchymal subtype is highly aggressive, it undergoes invasion, and thereby it is more difficult to treat than the epithelial subtype. Now we had a clinical validation where we had done. I'm just representing it with one slide. Through a collaborative effort with AFMC, with KEM, with TMC, we actually looked at around 97 cases of patients who had a large metastasis and who had a very poor prognosis. And we found that all three classes were definitely represented in this. Now, what we are trying to do is that since we understand EMT better than the epithelial subtype, because we've been working in the area of EMT since the last 20 years, we were able to develop a monoclonal antibody that we called as NAB150 that recognizes a surface molecule, annexin A2, on the surface of the mesenchymal cells, and it's cytotoxic in nature. And we actually screened it across a large number of cell lines, and we found that mostly it is very specific against only the mesenchymal subtype. So this was very satisfying because not only had we predicted that high-grade serous ovarian cancer is not only is not just a homogeneous disease, but you have different molecular subtypes that are not seen in histopathology, but which we could classify using a panel of 6 markers, and for which, and we definitely have some kind of a targeted therapy for one of those. So this is just a conclusion of all of what I've been seeing. Now, lastly, after we developed the monoclonal antibody, we saw that the monoclonal antibody, surprisingly, it reduced the tumor size. If you can see this, this is just one of the representative panels. This was done using a cell line we had developed, and the patient had passed away and was refractory to the therapeutic strategy in that patient was paclitaxel, cisplatin, and gemcitabine. So if you look at the drug profiles of these, it seems that this cell line is susceptible to paclitaxel, cisplatin, and gemcitabine. We also had been looking at several other drugs. Whereas the monoclonal antibody actually did not reduce the volume of the tumor very much, although the tumor stopped growing after a specific point of time. So this meant that there must be something about the residual cells that are present in the tumor. And it is very important not to look at just the volume of tumor that is lost following therapy, but to very closely evaluate the type of cells that are left behind, look at their positioning in the regenerative hierarchy, and then predict what is the likelihood that a particular or there would be disease recurrence after a certain therapeutic regime had been given to a patient. So we came up with this understanding wherein we were looking at the regenerative hierarchy, but also with the intrinsic host, the intrinsic tumor cell instability, as well as differential cell cycling to identify cells that are likely to stay just quiescent and then give rise to a recurrent tumor once the stress or the chemotherapeutic stress was removed. So what we came up with is the fact that there are at least a minimum of 18 different cellular subtypes within a tumor, and this is talking about a gross at a gross level. These are not identified with the help of any specific markers, but these have different regenerative capabilities. And then following treatment, one realizes that each, okay, so I'll just define these. The P18 defined the cancer stem cell component. What was very surprising for us, the aneuploidy component is very high in tumors, in ovarian tumors at least. And this, in fact, they can reacquire, they can undergo dedifferentiation and give rise to cancer or stem-like cells within the tumors. And aneuploidy actually imparts a kind of an evolutionary advantage to a cell by simply increasing the genetic component, some of which might contribute to the drug refractory behavior or drug resistant behavior. So what we understood was that when you actually look at this particular cytotype, as we call it, we found that the cytotype was differentially modulated in response to different treatments. And thereby we can come up with a very, okay, these are just the functional assays that we do for evaluating the regenerative functions. And thereby, if you actually look at it in this context, you can come up with a very specific drug combination. And with this could be a kind of a personalized type of a drug combination, which would achieve complete remission. We showed this in a batch of mice using a combination of 3 drugs. And what we found was that at the end of each of these 6 cycles, which is usually given to patients as well, what we had was the entire tumor burden was lost. The only cells that were left were in the differentiated component. We maintained these mice for over 6 months, and we found that there was no disease recurrence in this. At a molecular level, we understood that while the NAB 150 targeted the annexin A2 cells, because of the heterogeneity in the tumor, not all of the cells actually express the drug target. Similarly, gemcitabine could target only the actively proliferating cells for which RRM2 is the drug target. However, the use of a demethylating agent called as 5-AZA-DC actually mediated de-repression of the promoters of these genes, RRM2 as well as annexin A2, which were found to be methylated in several of the cells that actually did not express these molecules. So this combination worked over several cycles because at the end of each cycle, 5-AZA-DC would actually enhance expression of the drug targets of the other two drugs. So thereby, we could actually achieve a total remission in the mouse models. But as they say, a mouse is easier to treat than a human. The previous one?
So what was the model? What was the xenograph?
The xenograph was the A4, the tumor that the cell line that we had developed. We have done this actually using two models. We had developed a Pdx model, which was directly, so we took this specifically the mesenchymal subtype. We had already histopathologically identified it as a mesenchymal subtype. And then we developed a Pdx model and we used it in that. Along with that, we used the A4 cells, which I showed earlier, came from a patient who was refractory to gemcitabine, paclitaxel, and cisplatin. So we used the same model in these experiments. And this worked very well with both of those. But when we try to do it with the epithelial one, the part that is targeted by MAB 150 does not work very well. We did it with several other drugs also. So this is something that achieved total remission. Whereas for the epithelial subtypes, we are still evaluating drugs to find out what is the right combination that might perhaps work out in those particular types of tumors.
What is the percent distribution in the population?
So that differs actually in the different sites of metastasis. So the primary tumor is still, even in a mesenchymal subtype, I actually skipped those slides. What we had was a panel of 6 markers and we had a scoring system. So depending on and annexin A2 was one of the marker. So depending on the expression of all these 6 markers, that is how we actually stratified these tumors into either the epithelial, the mesenchymal, or the double positives. And in the mesenchymal subtype, the composition of the epithelial cells was less than 10%. And this was in the primary tumor. However, some of these, when they metastasize, they actually lose epithelial expression completely. So we have a loss of some of the markers, and E-cadherin is one of them, which makes us wonder how does the cell actually just grow and give rise to a tumor. But there are some of the other junction molecules that are expressed in such situations. Can I move on? So to wind up several of the stories which I have shared with you today, I think plasticity is the real challenge to us when we think about how we can treat a patient with. For us, we think only of ovarian cancer, but on a larger perspective, one has to think about how to achieve therapeutic efficacy in most of the cancers. So this is very complex and it involves a lot of complex issues. Even right now, we find that we can actually work efficiently only when we work in an individualized manner, and thereby comes in the feature of personalized cancer therapy. And that is something that we are moving towards in the future. These are just the publications. And yes, I think as I started off with saying that for me, this is a very creative. For me, I'm a simple researcher who just goes wherever research takes me. So that is my creativity. And I'm glad I have a bunch of very creative students as well. Now, I have always had them, but this is in the most recent batch, Ankita and Amruta, who are absolutely talented artists. And I'm glad they're doing research, because otherwise they would definitely be successful elsewhere. But they have just generated this for today's talk. And this is my current group. And I have several collaborators with which, especially the clinical collaborators, with who have actually enhanced our research. And the funding I receive is mostly from DBT, ICMR, DST, as well as the United States funds, and a lot of alumni who have contributed to several aspects of this story. I thank you for a patient hearing.
Thank you so much, ma'am, for sharing your lab's research with all of us. And now we'll take a couple of questions. We have a limited time, so yeah.
The plasticity that I've learned was an emotional intelligence, that how plasticity helps. But this was really interesting talk. I have one question on the trans differentiation that you have mentioned. You also said that it is involved in the cancer development, but has it been reported in vivo?
Oh, yes. So trans differentiation is something that has been reported way back, especially in the embryonal tumors. So you have embryonal tumors that are generated, even say in ovarian carcinoma, you have the germ cell tumors, and you have different components of, say, even teeth and nails that are present in these tumors, hair, teeth and nails that are present in these tumors. And that was the first, that is probably one of the first in situ reports of trans differentiation.
Another very interesting talk. Very basic question. So there are contrasting reports on stem cell fate, which say that stem cell fate is histographic, and some say it is niche dependent, and some say it is independent, and some say it's mixture of both. So my question was like one of the principles of stochastic fate and niche independence is the asymmetric. And one question which I had was like, is it possible that even in symmetric division of stem cells, there is some kind of a Paracrine communication which leads to between the symmetric stem cells, or one asymmetric and one differentiated and one self-renewing stem cell, which leads to the differentiation of that particular self-renewing stem cell, some kind of a Paracrine communication between them.
I think this is a very important question. You know, it's been very difficult to understand asymmetric division. Very often it's thought of as a metaphor, you know, one doesn't really understand about it. But over the last 2 decades or so, people have been understanding it. And even in the normal tissues or in normal organs, you have the niche that is very well defined. And the minute that niche is perturbed, you have a change in the distribution pattern of shift from asymmetric divisions to symmetric divisions. So we have been looking at this, and it's very difficult to get a grip on how do you define asymmetric divisions and symmetric divisions. So we came up with this thing, and it's actually documented by Sigma in its technical literature. What we did is that we labeled the cells with a vital dye, which it's a lipophilic dye, which labels the cell membrane. And then what we look for are the divisions in terms of dye dilution or dye quenching across different cell cycles. And we would harvest cells at different time points to understand how many of them actually would retain the dye, how many of them would undergo dye dilution, and how many would undergo complete quenching. Now, I didn't put up that study over here actually, but what we also did was that we injected these fluorophore tagged cells in mouse models, and we found that quiescence is something that is acquired only in vivo. We could never get quiescent cells in vitro. And probably if they were there, they would be diluted out by the more proliferative cells. So say this is something we published in 2009. It was a stem cell. Anjali was the student who was doing this work, and we went on to show that the in situ microenvironment, be it, you know, we are just doing subcutaneous tumors at that point of time, even in orthotopic tumors, wherein you have a specific niche that one would expect for a ovarian cancer, but even in a subcutaneous tumors, you had cells that would stay quiescent. We would sort out using flow cytometry, we would sort out these cells, bring them into culture, look at all the classical functional assays, or put them back into mice. And what we found was that cells that underwent complete dye quenching were the least regenerative, those who had a limited amount of dye left. So we had 3 quadrants in flow cytometry. And the middle ones were the progenitor, the ones with the highest intensities were the cancer stem cells. And accordingly, we could actually put a gradient, assign a gradient of regenerative potential to these. And we've actually inbuilt that into the model I showed you, the heat map, where we could predict the regenerative capability based on the amount of dye that was expressed. But that definitely suggested to us, at least in the system that we are working, the niche is probably the most important component.
Thank you, ma'am, for such an informative talk. I have a basic question. In addition of the transcription regulation, have you looked onto this epigenetic changes also? Because these kind of changes also play a very important role. And if these epigenetic changes can be used for development of personalized cancer.
Yes, so we have been looking at that, and one of the features from the epigenetic studies is where we used 5-AZA-DC. We have a target even for that. PTGIS is the molecule that we use as a biomarker to find out the extent of efficacy of 5-AZA-DC. And apart from, now the major problem with epigenetic drugs is that they have global effects. They do not have targeted effects. Like a monoclonal antibody would target only the annexin A2 expressing cells. Gemcitabine more or less targets only proliferating cells, especially those which express RRM2. So being a kind of a global type of having a global target actually helped us, because the promoter of annexin A2 and the promoter of RRM2 actually have a CpG island which is methylated in the cells that do not express the molecule. Within a tumor, you have, say, annexin A2 expressing cells and those that do not express annexin A2. By treating with 5-AZA-DC, what we did was that led to demethylation, as a result of which the cells which were not expressing annexin A2 started expressing it. And as you saw, we had 6 cycles of the drug, so that in the next cycle of the drug, cells that started expressing annexin A2 actually could be targeted. So this is the personalized therapy that I mentioned.
Thank you very much. That was fantastic. So I have a basic question. Although you said that in vitro you cannot see that, but in vivo still you start with a basic adult patient cell. So it may get accumulated with a lot of genetic mutations or whatever it may be. So how could you find it in vivo itself? You have a patient cell which is having a lot of mutations. That's the first question. And second question, that in these patient cells, don't you have any checkpoints to look at them? What are we doing? That's the second question.
Checkpoints for?
Whether you're P53 or anything mutated, like whether I'm getting more mutations or how to get rid of these cells, that's the second question. For the plasticity, basically, when we talk about whenever you're looking at the plasticity, probably you may need a lot of cell-to-cell communication is required. So do you, what kind of cell-to-cell communication do you have here? Because you're talking about personalized medicine here. Yes.
Yes. Very good questions. So to address your first question, we studied that in using the Pdx model. So within the Pdx model, we have one of the Pdx models that we developed. We took it through 15 generations. That is, we also established cell lines from I think P4 or P5. I don't remember exactly. So within that Pdx model, what we had is that we would just harvest the ascites from the first mouse and maintain it. Like you subculture cell lines, we would just subculture it through mice. And we stopped at P15, thinking that there are a lot of changes that would have occurred. At each level, we would also do RNA sequencing. We would have liked to do whole genome sequencing, but unfortunately, we didn't have the funds for that. But that was a very interesting study, because what we found is that the maximum changes from the human to the mouse actually happened from that transition, when there was a change in the species. But when you move from, say, P1 up to P15, although there were some genetic changes, they were not marked enough to actually count as being effective. So that is why we could actually use all these generations for evaluating the drugs. So that's as far as your first question goes. Secondly, you asked for the effects of paracrine secretions. Now, for doing that, we've done two things. We've done co-culture studies with stromal cells, wherein we tried to look at the effects on that, and we did fine. For example, we did it with stromal cells and also CD133 expressing cells, which we think are primitive endothelial stem cells. So we would treat them with mitomycin C, which would arrest their proliferation, and then do a co-culture of the tumor cells with these. And we found that to some extent, there was a modulation of the transcriptional factors. That is a study that was done by a master's student. Unfortunately, we didn't really take it ahead. But what Sagar did take ahead is that we also looked at the expression of these transcription factors with respect to, say, additives like EGF, BMP4. BMP4 was something we found was capable of converting a mesenchymal phenotype into an epithelial phenotype. And when EGF and TGF-beta converted the epithelial phenotype into that. So definitely there is a role for paracrine secretion in these mediating this type of a phenotypic plasticity.
Dr. Sharma,
Yes.
you opened my eyes to something I'm not very familiar with, but I want to ask you two questions. One is, how do you explain ovarian cancers being more aggressive and more common in both extremes of age, the younger and older women?
Yeah. I think that's a very important question. So the reason why ovarian cancer is very aggressive is that it's often called as the silent killer. If you notice, one of my earlier students made a kind of I don't know what to call it, a hologram for our group, and we say EOC, that is epithelial ovarian cancer silent killer. Very often, in fact, there is a public database called as the Cancer Genome Atlas, the TCGA as it is called, which it has above 500 tumors. I mean, the omics data of over 500 tumors, of which only 17 of them are in grade 1. I think 27 or 22 in grade 2, and the rest of them are mostly grade 3. Of all the samples that I have handled so far, none of them have been in grade 1 and grade 2. So it is a completely asymptomatic disease until it reaches, as our clinicians actually tell us, that mostly patients come to the clinic when they have accumulation of ascites, which becomes a feature that causes a lot of discomfort. And that is why they come to the clinic. They have no other symptoms sometimes, even at that stage. But they just think, most of them start thinking, oh, they're just putting on weight. And that is why it goes undetected until there is metastasis. And that is one of the reasons for its high mortality. Secondly, it usually does not, earlier it was thought that it is usually a post-menopausal disease, especially the high-grade serous one. Although the dysgerminomas, you have different histopathological subtypes of ovarian cancer. The dysgerminomas or the germ cell tumors are actually quite prevalent even in young girls. So teenagers also have dysgerminomas, but not high-grade serous ovarian cancer. So one does think it is the hormonal play that is a huge etiological contribution that could lead to some of these. Although increasingly with the lifestyle changes, our clinicians are coming up with younger patients in their 30s and 40s also who have EOC.
So that takes me to a very related question. So this plasticity, is it influenced by the hormonal level, the genetic predisposition, and physiological therapy?
As far as the genetic predisposition, it is shown that BRCA1, BRCA2 mutations. And in fact, what we found is that the BRCA1, BRCA2 mutations are present in our epithelial subtype, which opens up the possibility that they can be inhibited with PARP inhibitors, which are being increasingly are found to be effective, more effective actually in ovarian cancer than in breast cancer, surprisingly. but the BRCA1, BRCA2 mutations are a small component of the epithelial subtype, so suggesting that there are several other features that are still present. And we have a study ongoing on those because surprisingly, actually, you have only P53 mutations, BRCA1, BRCA2 mutations, and PIK3CA mutations that are actually more prevalent in ovarian cancer. The rest of the mutations are less than 1 to 2%. So it's kind of heterogeneous for us to pinpoint anything about that. So it is still more complicated, and we probably, even at the end of 20 years, I sometimes think I know lesser today than what I did when we started off. Ignorance was bliss at that point of time.
Yeah. So thank you so much, ma'am, and thank you to the audience as well for being so patient and so much into the discussion to take it to a really high level, to bring in new thoughts into the field as well. So, but our gastronomy department is really now pushing in to move for that. I request my
Just have 1 minute before we close with the just a minute. In fact, what we were talking this morning about our Institute Day, I had to go in between your talk, excuse me for that. It was our former DDP secretary, Dr. Rna Madam. She is so generous to call us all the way, and she's conveyed her best wishes to everybody, all NIABians, and in fact, we wanted to have her here today. And when I told that we have Dr. Priya and Dr. Sharmila with us, she was just telling them, oh, I missed it. But she was very emotional in conveying her best wishes to all of us. She repeatedly said, please tell. I said, yes, I will. So this is all, and I feel very happy and blessed that people, she was the one on 11th August, that time she was the secretary when Minister came and Plasticity was the director. Over to you.
now it's time to close, so I request my colleague to do the program.
Good afternoon, everyone. I will not take a minute. So, I know you all are handy. Respected Chief Guest, Director NIAB, distinguished professor, my colleague and students, I'm honored to give this vote of thanks on behalf of NIAB. We are very grateful to our Chief Guest, Professor P. Abhinam and Dr. Sharmila Bapat for accepting our invitation and taking time out and sharing with us and gracing this function. And a big thank you for their informative talks. I also thank the dignitaries and participants who joined online for this program. I also thank NIAB divisions right from administration, store purchase, finance, engineering, ICT, security, for their cooperation and support. Most importantly, students for their overwhelming support. And all my team members in the Scientific Event Management Committee for their help in making this program successful. Thank you.